Hi everybody, my name is Navarita Rai Chattopadhyay and I am serving as an assistant professor in the Department of Biotechnology, Haldia Institute of Technology. Today I will discuss a few things which are simply grouped as mutations. We all know that we carry the information for all our characters in genes, and genes are nothing but parts of DNAs which are packed with the help of some histone and non-histone proteins, and finally we get chromosomes. So simply we tell that chromosomes carry the genes and the genes are responsible for all our characteristics. Now if any sudden change occur in any of these genes or any part of a chromosome, then our normal bodily functions may be hampered. And such type of sudden changes are called mutations. Now mutations can basically be of two broad types. The first type, which occurs in the DNA level or molecular level, only 1, 2, 3 or a few nucleotides are changed within a gene, and then the function of that gene may be lost or may be initiated. If a gene like tumor suppressor gene should be active in our body and a mutation has occurred in such a way that the function of the tumor suppressor gene is lost, then tumors may occur inside our body and that is harmful. Here this loss of function is harmful. On the other hand, if any oncogene is mutated in such a way that is being expressed. Usually oncogenes should not be expressed in our body. If oncogenes are expressed, we may get tumors or eventually cancers. So oncogenes should not be expressed. But suppose one mutation has occurred in such a way that the oncogene has become activated, so it will be expressed. So mutations can either activate a gene or may suppress the function of a gene. Now in DNA level, if only 1 or 2 nucleotides are changed, then it is called a point mutation. Now the effect of that point mutation can be havoc. For example, in this image you can see that a start codon that is marked with a green one, ATG, and a stop codon which is marked with red. These two codons mark an ORF, that is open reading frame, which can code for a protein. Therefore, this part is the actual coding region of a gene. In that sequence, you can also see that just ahead of the open reading frame, one CG sequence is also marked with blue color. Now, if this base pair is replaced with a TA base pair, that is called substitution. No nucleotide or base pair is deleted, nothing is added, but just one base pair is changed to another base pair. So such type of mutation is called a substitution mutation. Now here I will like to mention one thing, that if a pyrimidine is changed with a pyrimidine and a purine is changed with a purine, such kind of substitution mutations are called transitions. Here you can see that the cytosine is replaced with thymine and guanine is replaced with adenine. That means pyrimidine to pyrimidine, purine to purine in that base pair. So this mutation is called a transition mutation. But sometimes it may occur that the cytosine of the earlier sequence is changed to adenine or guanine. That is, a pyrimidine is changed with a purine and the other one is changed accordingly. So here the purine to pyrimidine or pyrimidine to purine, that kind of change is marked as a transversion. So substitution mutations are of two types, transition and transversion. Now in the original sequence, the starting part was GA, TA, TA, AT, CG, then TC, TA, etc. Here in this image you can see that the G is missing from this part. So this is called a point deletion or simply deletion. In the next sequence, you can see an insertion. In between the C and the G, one C again is added. So this is called an insertion. All these mutations are still occurring in the DNA level. I'm not talking about the chromosome level mutations. One more thing, still now I have not shown any mutation within the ORF, that is the open reading frame. So the coding part, that is not changed yet. We know that genetic are triplets of nucleotides, and these triplets can code for one amino acid that is reflected in the mRNA. In mRNA, the same strand of DNA is exactly copied. Only in place of thymine, we can see uracil. In mRNA, these triplet nucleotides are called as codons, and that is called code in the gene. Here in the open reading frame, each triplet will mark the code for one amino acid starting from ATG. ATG codes for methionine, which always is the first codon or starting codon. Although there are a few exceptions for bacteria where you can get isoleucine instead of methionine as the first amino acid, but discussion on that is beyond the scope of today's discussion. So come back to ATG. ATG codes for methionine. And the last one is TAA, that is a stop codon. So it does not code for any amino acid. Now let me show you if any mutation occurs within the reading frame, that what may occur. Here in this image, you can see if any substitution mutation occurs within that peptide code, the amino acid may get changed. Here in the lower part, I have shown one insertion. That is a point mutation with just one nucleotide inserted. So here you can see that the frame for protein production is completely changed. It may start with ATG, but it cannot read TAA directly as a codon, and it cannot stop. So eventually it will not produce any protein. So these type of things are called as frame shift. So the frame for the protein production which was present in the upper part, as you can see starting from ATG, if you take triplets of nucleotides, you will get 6 amino acids for your polypeptide chain. In the lower part, that frame is changed. So it will have limitless amino acids as it cannot get the TAA triplet in its reading frame. This type of mutation is called a frame shift mutation. Now remember one thing, that is frame shift mutations occur only when one or two nucleotides are added or deleted from the DNA of the open reading frame. If 3 nucleotides are added or deleted, that 3 will code for another amino acid. So the frame will remain same. That will not be called as a frame shift mutation. So for frame shift mutation, only 1 or 2 nucleotides are added or deleted. Now the next is inversion. Here a small part of DNA is cut out and is inverted and then joined again. This is called inversion. Again, it may hamper the protein production because as it will start reading from ATG, the amino acids may become changed. The reading frame may become same. So you can get an what if. But the amino acids which were coded by the original sequence is changed in the new sequence with the inverted region that may code for a few new type of amino acids or altered type of amino acids. So eventually the final protein will be altered. Now 3 more types of mutations. Nonsense mutations. This type of mutation will create a stop codon. So it will stop the protein production. Missense mutation. In missense mutation, the nucleotides will be substituted in such a way that the amino acid may be changed, but the new amino acid will be biochemically similar to the previous amino acid. Say for example, the amino acid was lysine in the previous gene, in the normal gene. So lysine is a positively charged amino acid. If the substitution mutation occurs in such a way that instead of lysine, histidine is present, so it will not hamper the overall form of the protein because histidine also is a positively charged protein. The effect will be somewhat neutralized. And the last one is silent mutations. In this case, the insertion, deletion, substitution, everything occurs outside the gene. So it will have no effect on the gene. Sometimes it may occur within the gene also, but that will have no effect. For example, some amino acids like leucine have more than one codon, If one nucleotide is substituted in such a way that code for leucine is altered, but again it has created another code for leucine. So finally you will get leucine amino acid just like the original protein. So such kind of mutations will actually have no effect on the protein production. This is called silent mutation. Now chromosomal mutations. Chromosomal mutations involve larger parts of DNA, that is larger parts of chromosomes. So it will have some impacts on the structure of the chromosomes and eventually will affect the individual. These are also called as structural aberrations. Basically these are of 4 types. One is deletion. A part of chromosome is deleted. Within this part a whole gene or more than one gene may be deleted. Next is duplication. A part of chromosome is duplicated. So one or more genes may become duplicated. Next is inversion. Just like the inversion I have told about DNA mutations, here also a part of chromosome is cut out and is inverted and then joined again. Now here the genes may remain intact, but the problem is during cell division, chromosome pairing is hampered. Now if chromosome pairing is hampered, their separation in 2 daughter cells will also be hampered, and that may result in some numerical aberrations I will tell later on. Finally, the 4th type of chromosomal aberration or structural aberration is translocation. Here a part of one chromosome is cut out and is translocated, joined to another chromosome. So again during cell division the chromosome pairing is hampered. Now comes another type of chromosomal mutations which is called numerical aberrations or ploidy. We know that all our chromosomes remain as pairs. So normally 2 members of chromosome 1, 2 members of chromosome 2, 2 members of chromosome 3, like this all the chromosomes are paired up. In case of sex chromosomes, 2 X chromosomes are present in female and 1 X and 1 Y chromosome are present in males. Except of the sex chromosomes, all the other chromosomes which are called as autosomes, the members of each pair contain exactly same genes in the same position or same locus, but the alleles may differ. Say for example, in one chromosome pair, the gene of height is present in these 2 lines. Now this member may contain the tall allele and this may contain the short allele. Both of these may contain tall allele. Both of these may contain short allele. So the gene of height will be same in these 2, but the alleles may vary. This rule is applied for all the autosomes. The genes and their loci will be same in the members of the autosomes, but the allele may differ. That's why we call that our genome is 2n number. N is one set of chromosomes. If you take one member from one pair and make a set, that will make one ploidy or monoploidy, or simply we denote that as n. We have 46 chromosomes, so we have 2n numbers of chromosomes. Almost all the normal eukaryotes carry 2n numbers of chromosomes. The total number of chromosomes may be 4 or 6 or 8 or whatever it is. In humans it is 46. The point is the total number will carry 2 into n number of chromosomes. If this number varies, if any change from this number is seen, that is called a numerical aberration. Numerical aberrations can again be of 2 basic types. One is called euploidy and the other is called aneuploidy. In case of euploidy, the total n number of chromosomes are either duplicated or are deleted. Say for example, if 2n number of chromosomes are duplicated and an organism has generated which have 4n number of chromosomes, that is called tetraploidy. That means 4 into n number of chromosomes are present. If 3n numbers are present, then it is triploidy. Likewise, if a single n number of chromosomes are present, then it is called monoploidy or haploidy. In normal condition, we can see monoploidy only in gametes, but that is not seen for normal individual. In normal individual, the total number is 2n number of chromosomes. And And this normal condition is called diploidy. So euploidy can be of monoploidy, triploidy, tetraploidy, pentaploidy likewise. Now the next type of numerical aberration. One is called hypoploidy. That means one or more number of chromosomes will be less than the normal twin number. That again can be of two types. One is called monosomy, when only one member of one pair is missing from the whole twin set. Then it is called a monosomy. Now suppose two pairs are there. One member from this pair is missing. One member from this pair is also missing. This condition will be called as double monosomy. Likewise, triple monosomy may also occur, but obviously that individual will not survive. The next variety of hypoploidy is nullisomy. In this case also, the chance of survival of the individual is almost nil. The next type of aneuploidy is hyperploidy, when one or more number of chromosomes are added as an extra chromosome with the total 2n set. Now suppose this is chromosome pair 21. If another chromosome 21 is added, then these three members present as chromosome 21, they will be called as trisomy 21. A very good example for that is Down syndrome. I will discuss later on. So one member extra with the normal two members, that is called trisomy. If two members are extra, then it is tetrasomy. If two pairs of chromosomes we are considering and each of these will contain one extra chromosome with them, then we can call that as double trisomy. But again, the chance of survival is very low, almost nil. Now how these numerical aberrations occur? The process is called as non-disjunction. When gametes are formed, the egg precursor and the sperm precursor, both of them will contain the normal 2n number of chromosomes. One member from each pair will go to one gamete and the other member of that pair will go to another gamete. If this process occurs properly, we normally get 22 autosomes in one gamete and one sex chromosome in that. In the other gamete, another 22 autosomes and the other sex chromosome will be present. That is the normal condition, and that process is done by the help of formation of spindle fibers. Now sometimes the formation of spindle fiber may be hampered by some processes, some biochemical conditions, or simply by chance. In that case, if both the members of one pair move to one gamete and none of them have moved to another gamete, then the process is called non-disjunction, and one gamete will have one extra chromosome and the other gamete will have none of that. So we will get abnormal gametes, one with 24 chromosomes and the other with only 22 chromosomes. Now I will discuss a few examples for such type of non-disjunction and related aneuploidy. The very well-known one is Turner syndrome, where one X chromosome is missing in the female. We all know that when both the X chromosomes are present, the female is called a normal female. If one X is missing, that female is called as a Turner female. It is named after the scientist who have discovered this. The Turner females will suffer from various structural and mental problems. They will have shorter height, impaired breast development, rudimentary ovaries, no menstruation, etc. Obviously, these are infertile. The next one is Klinefelter syndrome. Here, it is a male. Again, the name is given after the scientists who have discovered that. Here, two X chromosomes are present and one Y chromosome is present. So, normally the male contains one X and one Y. But here, an extra X chromosome is present. This person will also suffer from various problems, like some female-like characters like breast development, female-like pubic hairs, long arms and legs, small-sized testes, and poor beard growth, etc. They will also suffer from a little bit of mental retardation. Next one is Down syndrome, again named after the discoverer, and it is also called as trisomy 21, as one extra member of chromosome 21 is present here. these children face severe problems related to their structure, growth, and development. They will also suffer from mental retardation, and generally their lifespan is much lower compared to normal human beings. That's all for today's video. If you have any question, you can write me in the comments. If you have liked my video, just press the like button. Stay healthy, stay safe, stay tuned. Bye.